All right, everyone, 3 o'clock, let's get started. I am recording the review session. Hopefully my voice lasts in this next couple hours. Okay, so I'm really hoping that I can get through this. Okay, so does anyone have any questions right up front? Like something that you were really confused about that you just want to clarify right away? Pretty good? You just want to start from the beginning? All right. Okay. So the first thing, as we've talked about before, is just knowing the general anatomy. I do know that students get ureters and urethra mixed up all the time. So just make sure that you're clear about those terms. The kidneys are those bean-shaped organs that are more towards your dorsal side, towards your back. And technically, these kidneys are retroperitoneal, which actually means that it's outside the peritoneal cavity. The peritoneal cavity actually kind of holds all of your organs in your abdomen in one sac, basically. The kidneys are actually outside that sac, the peritoneal cavity. The other thing that I really want you to know is the general anatomy of the kidney itself. Okay, so what you have here are the major difference, do you know this, between the cortex region and the inner medullary region, the medulla. So the cortex is this outer part here, the lighter shaded area, really towards the outer capsule of the kidney. And the medulla is, we had this around Halloween, is that pyramid, this pyramid structure right here, the darker pyramid structure. So people that have the lat were actually able to do a cross-section of the kidney, and you could very easily see the medullary region, okay? Then just wanted to make sure that you all are aware of the minor and major calyces. The minor calyces is right here by the renal papilla. That's the very end of the inner medullary region. It's called the renal papilla right here. And then the cavity right next to the renal papilla is called the minor calyx. Plural, that's calyces. Okay? Then this larger cavity right here is called the major calyx. And then you have this renal pelvis area that then allows for the urine to enter into the ureter. Okay? Now, remember, the blood supply is really important too, because the kidney is all about filtering your blood. So just notice the renal artery here, depicted in red, and the renal vein. Okay? All right. Here's just another picture of it, maybe easier to see. You've got the medulla, that's the medullary region. Remember, we've talked extensively about that osmotic gradient. So make sure you realize that in this region right here, at the top of the medullary region, is only about 310 milliosmoles. But here by the renal papilla, it can be as concentrated as 1,200 milliosmoles. Okay, so renal papilla, minor calyx, major calyx, renal pelvis is here, and then the ureter. Pretty good with the anatomy? Not too bad? Okay, so this is an enlarged nephron, just to make sure. Remember, there's about 1 million nephron within each kidney. So that is not to scale, is what I'm trying to say. All right, so then one of the things we talked about was the difference between the cortical nephron and the juxtamedullary nephron. So there's two types of nephron that I just kind of featured. The cortical nephron really is more or less in the cortex region.
Sorry, we got lost.
Oh, no worries. I hope this room is, the only thing that's really bad about this room is, I forgot about this, there's very little whiteboard space, And if you want more, you actually have to lift up the screen. So hopefully I can get everything in this whiteboard that I want to. So we were just going through the anatomy of the nephron, just major anatomical features. Yeah,
Thank you.
yeah. Major anatomical features. And I detailed two different types of nephron, the cortical nephron and the juxtamedullary nephron. So notice that the cortical nephron is mainly contained in the cortex region, in the outer region, and it has a very small loop of Henle. So realize that the cortical nephron does not participate in setting up this osmotic gradient in the medullary region. That's really due to these juxtamedullary nephron, okay? And these long loops of Henle. Those long loops of Henle are what are responsible for setting up that osmotic gradient in this entire region. Okay, pretty good? All right. Also, these are the paracapillary paracapillary vessels right here. And we talked about this pretty extensively in the lecture. This is the vasa recta. Okay, so this is the blood supply that surrounds the juxtamedullary nephron. And the vasa recta is responsible for maintaining that osmotic gradient that is set up by the juxtamedullary loops of Henle. Okay.
More like surrounded and like
Yeah. So yeah, that's a really good point. I showed you I actually showed you later on, I'll find it, but essentially it wraps around much like these vessels. Yeah.
So just for diagram purposes, to like separate.
Yeah, exactly. So we also talked about countercurrent systems. And just know that the loop of Henle is called a countercurrent multiplier because remember, it actually uses active transport mechanisms in order to set up that osmotic gradient. It's really the sodium potassium 2-chloride cotransporter and the pump that is pulling salt out to actually set up this gradient. Yeah.
When nutrients are reabsorbed into the blood, are they with the fluid in that?
Yeah, so when we say reabsorption, it means we're pulling molecules out of the nephron. They're going across the epithelial cells through this interstitial fluid and then entering into the vasa recta. Okay, that's reabsorption. Question?
In the cortical nephrons,
Yep.
is it still called physiology?
No.
What's it
Okay.
called?
Paratubular capillaries. Paratubular capillaries. So here's another thing. The vasa recta is very specific to the juxtamedullary nephron. And the vasa recta is called a countercurrent exchanger, not a multiplier, because it really relies on just passive properties. No energy is needed, right? So I'll detail that in just a little bit, but as the red blood cells and the fluid flow down towards the intermedullary region, it's going to, salts are going to enter into the vasa recta and water's going to be pulled out. But then as it kind of goes back up towards the cortex region, the water is actually going to be entered back in and salts are going to be pulled out, right? So basically you're going to see that it's going to be very concentrated at the end of the loop, and then it's going to be more like 325 milliosmoles at the top. All passive properties, and we'll go over this in a little bit too, but these are all passive properties. No energy is required. It's basically just diffusion and osmosis. Okay? So countercurrent multiplier versus countercurrent exchanger. And the analogy I used for the vasa recta was the analogy with heat transfer in the arm, right? So as blood enters into the arteries into your arm, it's very warm. As it travels down towards your fingertips, it cools off. But because the veins and the arteries are so close together, as the blood returns in the veins, it's so close to the arteries that it actually warms the blood. So there's not much of a temperature difference once that blood enters into your core. It kind of helps to maintain heat within your core region. So that's very analogous to what's happening with the vasa recta. Okay, pretty good? All right, so again, as you go down, salts are entering in, water's being pulled out. As you go back up, water's entering in, and salts are being diffusing out. Okay? All right, so keep going.
Why is there a dog?
2 of them.
Oh my god.
They're visitors. They want to know more about physiology.
He's only 6 months, he can't be home alone.
Yeah.
Oh my goodness.
I love puppies, so I don't mind at all. You should see in my orientation, I made the announcement that I love dogs. We have the dogs in Bailey Hall too.
Oh, the fetch dogs.
The fetch
I had one
dogs,
last year.
and other people are bringing their dogs. I, at any one time, will have like 5 or 6 dogs in that class. Yeah, it's fun. And so just keeping them.
Yeah, I had Turbo in your class last
Oh,
semester.
yeah, that's right. You brought Turbo. That's right. All right, so moving on, keeping going here. As long as my voice is going to hold out here. Here's the tubular tour, right? I do want you to know what's happening in each of these segments, okay? So we have the proximal tubule first, the loop of Henle, which includes the thin descending loop, and the thick ascending loop. Okay, so this is the ascending loop, okay? Distal tubule starts here. Don't forget about tubuloglomerular feedback, right? We'll talk more about that intrinsic pathway in just a second. Distal tubule and collecting duct. Okay? All right. If you don't remember anything but this in the proximal tubule, it will help you. The proximal tubule is all about reabsorption. Okay? So one word really encompasses the proximal tubule. It is reabsorption. So what happens is blood enters into the glomerulus by the afferent arterioles, and these glomerular capillaries are under super high pressure. It forces plasma out, right? The larger blood proteins like erythrocytes, red blood cells, right? And albumin, they stay in the glomerular capillaries, right? But smaller molecules, ions, glucose, amino acids, they're filtered out. So that ultrafiltrate, that's what I'm going to call it, or primary urine, both the same term. It enters into Bowman's space, okay? This whole thing is called Bowman's capsule, but this space right here is called Bowman's space. And then it enters into the proximal tubule, where you want to recover all of your essential nutrients. That includes glucose and amino acids. So 100% of your nutrients are reabsorbed. 85% of your bicarbonate, very important buffer in your blood, is reabsorbed, recovered, brought back into your circulatory system. 70% of sodium and potassium, and 70% of water. Okay? All of that is being pulled out of the nephron and brought back into your blood. Okay? All right. So then the ultrafiltrate enters into the thin descending loop. This is where a lot of the solutes are equilibrating. It's going to enter into an environment that's very hyperosmotic, right? And solutes are going to go into the thin loop here, and water's going to be pulled out. All right? So water flows out, solutes equilibrate. It goes towards the ascending limb, enters into the thick ascending limb. This whole blue area here is called the diluting segment. The diluting segment. And why that is because water is actively being pulled out. I'm sorry, not water. Salts. Sorry, salts are being actively pulled out by the sodium potassium 2-chloride cotransporter. So you're pulling salts out. It actually pulls more salt out in the beginning than it does at the top. So you're starting to really establish that osmotic gradient within this entire region. You pull salts out, right? But water's not allowed to follow. So water's not allowed to follow. There's no aquaporin channels. It's very low water permeability. As you reach the top, that ultrafiltrate is now so dilute, it's only 150 milliosmoles. Okay? So just to clarify, because I had a moment there, salts are being actively pulled out, but water is not allowed to follow. Okay? In that diluting segment. Question?
Is it essential for us to understand the baseline levels of osmolarity for each stage?
Not really.
Just
Yeah,
understand.
yeah. I would say the main take-home message is in the very inner medullary region, it can be as concentrated as 1,200 milliosmoles. And then up at the top, it's usually about 300. So there's this grade of osmolarity all the way down. The reason why it's important is because on the back end, the collecting duct, this is what is giving you the capability of concentrating urine. So knowing this region down here is 1,200 milliosmoles gives you a basis for why the urine can be concentrated to 1,200 milliosmoles. Does that make sense? Okay. Questions? Any other questions? All right, great questions. So then the distal tubule and the collecting duct, there's a lot of similarity between the two. This is where sodium chloride continues to be reabsorbed, especially in the face if aldosterone is on board, right? Aldosterone really enhances that sodium chloride reabsorption. Also in the distal tubule and in the collecting duct, potassium and proton secretion. Now, when you actually look at the transport properties, it really doesn't go the other way around, right? You're not getting a whole lot of potassium reabsorption, or you're not getting at all proton reabsorption. Doesn't happen, right? We'll look at those transport properties in a minute. And that's going to be important with the acid-base regulation. That's going to be your lecture for next Wednesday, okay? Bicarbonate and bicarbonate here in both the distal tubule and the collecting duct continues to be reabsorbed. But the main thing here is when vasopressin is circulating throughout your body, it's going to promote water reabsorption and urea reabsorption. Don't forget about urea, because if you're reabsorbing a lot of water in this area, you're going to dilute out your osmotic gradient. So urea is an important osmolyte because it helps to maintain that osmotic gradient when you're really reabsorbing a lot of water. Okay? When you reabsorb water, you concentrate urine. Makes sense, right? All right. Any questions? So just remember with vasopressin, when vasopressin is circulating in your blood, it's going to enhance water permeability or water reabsorption, and you're going to get an increase in urea reabsorption. All right, sweet. Some people, if you want This table helps kind of summarize everything that I just talked about. And I did mention this comparative physiology example. Essentially, if you're talking about aquatic animals, I would even say frogs are a good example. They have no loop of Henle, none. There's no reason because they don't ever need to concentrate urine. Right? They live in an aquatic environment. But if we're talking about a kangaroo rat, that is probably one of the animals on this planet can really concentrate urine and conserve water. You can see it has the longest loop of Henle, the most pronounced renal pelvis, right? Because it has to accommodate these long loops of Henle, so that this animal can really concentrate urine and conserve water. Okay, so the major innovation is the loop of Henle allows the production of a concentrated urine, and animals that produce more concentrated urine have longer loops of Henle and thicker medullary regions, a more pronounced renal pelvis. Pretty good? All right. So our 4 different processes: filtration, reabsorption, secretion, and excretion. Now, I kind of made the analogy here, what's the difference between secretion and excretion? Because I got a couple of questions about that. And I said secretion is like throwing something into the trash into your kitchen. And then excretion is taking the trash out of the house. All right? So secretion is when molecules are delivered from the blood into the nephron. And then excretion is when that is eliminated from the body. When you basically urinate, micturition is another fancy name for urination. Reabsorption is when you're pulling things out of the nephron and delivering it back into the blood. You're recovering it. And then I just did this backwards. Filtration is basically at the glomerulus under high pressure, forcing fluid out into the nephron. Okay, filtration, reabsorption, secretion, and excretion. Okay, so we started off with filtration. This whole thing right here is called the glomerulus. It's also called Bowman's capsule. This space right here is called Bowman's space, and then these are the glomerular capillaries, okay? Blood enters in through the afferent arterioles, that brings blood to the glomerulus. Then it's filtered in the capillaries, glomerular capillaries, and then the blood that's remaining leaves via the efferent arterioles. So, if you remember from last exam, these capillaries are called fenestrated capillaries. They actually have these filtration slits called podocytes. And these podocytes can actually be controlled by the intraglomerular mesangial cells. Intraglomerular mesangial cells. These mesangial cells are not only here, you would call this extraglomerular mesangial cells, but they're also present close to the podocytes, and they can control those filtration slits. Mesangial cells can also close off these loops so you don't get blood into this area here. It lowers the surface area of filtration and basically can lower glomerular filtration rate. Make sense? Okay. Juxtaglomerular cells, these are the cells that actually release renin in response to low blood pressure. And then you have smooth muscle that surrounds the afferent arterioles. This is part of the distal tubule. I had someone in my office who was a little confused by this figure. What you're looking at is basically, I wish I had a stick. Do I have something that I'm too short? If I took a box and I just cut off and I was just looking at this area right here, what's missing is the rest of the proximal tubule, the loop of Henle, right? So we're only looking at this section right here, right? So that's what this represents, okay? This is the distal tubule, and the macula densa cells are detecting the flow through that area. So you're here at the review session. Hopefully people are listening. Be prepared to label a diagram that looks like this, okay? All right, this actually says a lot. It actually gives you a lot of detail about some of those intrinsic and even extrinsic mechanisms with Renal. So know the anatomy. All right, so the important message here, we're going to revisit net filtration pressure. If you haven't found the equation sheet that'll be stapled on the back of exam 3, it is in the respiratory review session. Okay, the respiratory review tab, basically. You'll find the exact equation sheet that's going to be stapled on the back. And net filtration pressure will be on that sheet. Okay, so we've learned this before. You should know what glomerular capillary hydrostatic pressure means, Bowman space hydrostatic pressure, and what is oncotic pressure, right? Oncotic pressure is referring specifically to the osmotic pressure of the larger blood proteins, okay? Now, the reason why there's no oncotic pressure in Bowman space is because larger blood proteins don't enter into that area. So there's 0. You can just take it out of the equation. So this kind of sums it all up. The glomerular capillaries, they have a hydrostatic pressure of 60. That is twice as much as in the vascular, any other place in the circulatory system in the body. Usually in the body, it tops out at maybe 36 millimeters of mercury, but in the glomerular capillaries, it's 60. That's actually pretty significant. It ensures that filtration is always occurring in the glomerulus. You have to subtract off the hydrostatic pressure in Bowman space, right? It gives you that net. And then you also have to subtract off the oncotic pressure in the glomerular capillaries, but again, not in Bowman space, because the oncotic pressure in Bowman space should be 0, unless there's some kind of disease state where larger blood proteins are escaping. You should never have blood in your urine, right? So if blood is actually entering into Bowman space, that's a problem, okay? It'll disrupt us. Now, the reason why the net filtration pressure is still 16 is you still have to have a pretty high pressure to make sure that the fluid goes through the entire nephron and then out through the minor calyx, major calyx, and then out through the ureters. This pressure right here, 16, is to ensure that the fluid keeps flowing through the nephron. Fluid's always going to go from high pressure to low pressure, Ohm's law, right? So net filtration pressure. In a minute, we're going to talk about these 3 intrinsic pathways. Let's see, but I do want to go through some of these particular figures that we've talked about before. Some people have an intuition about it. This is just about plumbing. I'm just telling you, this is just a plumbing issue. So when you constrict the afferent arterioles, you're going to actually reduce the hydrostatic pressure in the glomerular capillaries, which essentially reduces glomerular filtration rate. Remember, glomerular filtration rate is just flow. It's just a flow in mils per minute. So that's one way. You can expect a question like, which one of these is going to increase glomerular filtration rate? Which one of these is going to decrease glomerular filtration rate, okay? So knowing this plumbing issue will really help you with those types of questions. If you constrict the back end, kink that hose, right? If you constrict the efferent arterioles, everything upstream is going to increase in pressure, right? So you're going to increase that glomerular hydrostatic pressure in the glomerular capillaries and increase glomerular filtration rate. Stop me if I'm going too fast. Question? No. Okay. And then this, to me, when I was a student, was a little less intuitive. Okay? So knowing, you can kind of work backwards in some ways. If you now dilate the efferent arterioles, it's going to be the opposite of constriction. So you're going to decrease the hydrostatic pressure in the glomerular capillaries and decrease glomerular filtration rate. Okay, and then if you dilate the afferent arterioles, you're going to bring more blood into the glomerular capillaries. It's going to increase the hydrostatic pressure in the glomerular capillaries and increase glomerular filtration rate. Pretty good? Okay, so there's a question or two about this. Just letting you know, right? I really want you guys to know this material, so that's why we're going through it again. All right, again, the 4 different processes. This diagram has been distilled down to probably the most simplest form, right? So you've got the glomerular capillaries right here. This red part is the blood supply, and this yellow has been distilled down to the nephron. This is the filtration unit. This whole tube right here is supposed to represent proximal tubule, loop Van Lie, distal tubule, and collecting duct, right? And then excretion. So filtration happens at the glomerulus. When you pull molecules out, like vitamins that are maybe too big to be filtered, when you pull them out of the circulatory system, deliver them into the nephron to ultimately be eliminated, this is called secretion. Okay? Reabsorption is the opposite. It's pulling molecules out of the nephron back into the capillaries or the arterioles, capillaries, and then excretion is eliminating that urine from your body. Pretty good? I stress this because I see it a lot. Mistakes are made, and I know that you know the material, but I'm emphasizing this because I want you to know it, right? You should be prepared to actually see questions like that. So as I said before, with reabsorption, remember it's going across the epithelial cell layer into the interstitial fluid and then into the blood supply. All right. So, which one of these is filtered at the glomerulus, strongly secreted, and excreted? Is it X, Y, or Z? X, right? Now I'm going to put some things together for you. This is PAH. Remember PAH? PAH is a molecule that you can use when you plug that into the clearance equation. You're basically calculating, when you're calculating PAH clearance, you're calculating renal plasma flow. And the reason why you can do that is because PAH is freely filtered and strongly secreted, so that everything that enters in through the renal artery ends up in the urine. Nothing goes on to the renal vein. That's why this is a good marker for renal plasma flow. So you can test somebody's blood, figure out how much PAH, what their concentration of PAH is in their blood. You can then look at their urine, figure out what the concentration of PAH is in their urine. And when you plug that into the clearance equation and calculate PAH clearance, you're basically calculating renal plasma flow. All right, so taking a little different twist here, let's just say this is filtered and then not secreted or reabsorbed. So pretend this is whatever's filtered just ends up in the urine, not reabsorbed. This is a good example of creatinine, right? Creatinine is freely filtered but not reabsorbed or secreted. So whatever's filtered ends up in the urine, right? That's why this is a good marker for glomerular filtration rate. Does that make sense? So the clearance equation will be on the back of your exam. You will see it right away. If you calculate creatinine clearance, you're basically calculating GFR. If you're calculating PAH clearance, you're essentially calculating renal plasma flow, right? Because renal plasma flow is not only what's filtered, but what goes on into the efferent arterioles. All right, so filtration fraction is GFR over renal plasma flow, and it's usually about 20%. That's normal. Yeah.
I was just wondering, I was looking on like their review slides and it had links to the homework assignments and then it said like answer keys.
Yeah.
But they haven't been posted.
I just posted them.
Okay.
Yeah.
Okay.
So take a look at them again. I did get an email from another student.
Okay.
All of the answer keys, even for the review questions, they're all posted now. Okay, so you'll have them for the weekend. Pretty good? All right, so here's where we're really zooming in on those epithelial cells, right? Essentially what you're looking at here is the proximal tubule. And the reason why you know that is this co-transporter right here. Remember, the proximal tubule is all about reabsorption. So what happens here is glucose enters into the cell. It's actually transported across the basolateral membrane, delivered into the blood, right? This glucose could be, you could actually exchange this for amino acids. So also, amino acids are brought into the cell by the sodium amino acid cotransporter, and then it's transported across the basolateral membrane through this facilitated diffusion transporter. Sodium is reabsorbed. Potassium in this case is also reabsorbed. Some of it is actually secreted. The major thing also that I want you to recognize is for every carbon dioxide molecule that enters into this epithelial cell of the proximal tubule, it's converted to carbonic acid and then almost immediately converted to protons and bicarbonate. And because of the way these transport properties are set up, for every carbon dioxide molecule, one proton is secreted and one bicarbonate is reabsorbed. It's never the other way around because you don't have these sodium bicarbonate exchangers on the apical membrane. Okay, so one proton is secreted and one bicarbonate is reabsorbed. All right, next one. You should be able to recognize right away that this is the thick ascending limb. And the reason is this: sodium, potassium, 2-chloride cotransporter. If this is the inside of the nephron, you're pulling all of that salt into the cell and it's being delivered into the interstitial fluid. Okay? This is an active transport mechanism because of the pump. You need to have ATP to run the pump to make sure you're transporting sodium across the basolateral membrane. That's why in the thick ascending limb, this is called the counter current multiplier because of these active transport properties. So remember, this is the segment that's responsible for establishing the osmotic gradient. You're pulling all this salt out, mainly in the beginning of the thick ascending limb, and it's establishing that profound osmotic gradient within the medullary region. Okay? What's missing here is this reaction, right? But it's the same thing. It's happening in the thick ascending limb. They just took it out for simplicity's sake. Okay? And that's about it for this one. This is really all about salt reabsorption, but there's no aquaporin channels, so water can't follow that solute movement. That's why the thick ascending limb is the diluting segment. Okay? All right, now this looks like the easiest one, but it's probably one of the most difficult. Yeah?
I just have a quick question.
Yeah.
In terms of what would be best to remember from these slides, would you recommend being able to distinguish one
Yeah.
from the other?
Yeah, I would say one from the other, but it also gives you some insight as to how each of these segments work. So the reason why I kind of go into detail on it is because you can imagine like what's happening in these different segments and why is that osmotic gradient being established? Why doesn't it happen in the proximal tubule? So I would say it's just more or less helpful in really understanding the function of these different segments. Okay. Remember, Lasix is a molecule that inhibits this. So what would happen if you inhibited it? All of this salt would stay in the nephron, and it would be delivered to the collecting duct, where it would disrupt the osmotic gradient, and you would have a difficult time concentrating urine. So this is probably one of the most powerful diuretics, right? Lasix, we talked about that with horses, racehorses. The reason why this is so powerful is because you get this enormous amount of salt reabsorption, and if you prevent that, all of the salt gets delivered to the collecting duct. And again, you diminish that osmotic driving force for water reabsorption when vasopressin is present. Okay? Do you want me to write that out? I see some like, how does that work? Okay, so, okay, so if you have the loop of Henle here, and the distal tubule, and then you have a common collecting duct. Okay? So what usually happens is you are pulling salt out more in the beginning of the thick ascending limb. Okay, with me? So this is establishing that osmotic gradient. If you block that with Lasix, what happens to all the salt? It actually stays inside and then gets delivered to the collecting duct. Right? This is very hyperosmotic down here compared to up here. Okay, so before I deliver all that salt, if it's working normally, remember how we talked about osmosis? When you look at this blank area, you should automatically think, oh, this is high in water molecule concentration. And water's always going to move from high concentration to lower concentration. So as fluid moves down this collecting duct, it's going to encounter a more and more hyperosmotic environment, and you're going to be able to pull more and more water out, right? So now if you inhibit that sodium potassium 2-chloride cotransporter, you're going to deliver all of the salt in the collecting duct. You're going to lower the water molecule concentration, and now you're not going to be able to pull as much water out. So you're not going to be able to concentrate urine. That's why it's a very powerful diuretic. So what's going to happen with Lasix? You're actually going to enhance the excretion of sodium, potassium, chloride, and water. You're going to enhance the excretion of all of those things. Pretty good? It is the most powerful diuretic out there, Lasix. It's also called furosemide or bumetanide. But you can imagine, not only are you losing a lot of water, you're disrupting your electrolyte balance too, because you're losing a lot of salts, right? So keep that in mind too. Okay, so here is where we are at the collecting duct, and I'm gonna bring in aldosterone. So what we're doing now is we're actually going back to the intro to renal physiology, but because you know so much now about aldosterone, I'm layering it on right now. You know the clearance equation. You actually know renin and aldosterone now. So now this may be hopefully starting to make a little bit more sense than when I first introduced it, okay? So aldosterone is a steroid hormone that can easily pass across the plasma membrane, enter right into the nucleus, and what it does is it promotes protein synthesis of ENaC channels. ENaC. I'll just put it over here because it's part of the collecting duct. ENaC, epithelial sodium channel. Yep. Okay, so you increase the number of ENaC channels. What that does is it promotes more sodium reabsorption. When more sodium enters into the cell, it increases the turnover rate of the pump, bringing more potassium into the cell, and then you get enhanced potassium secretion. Okay, so it makes sense if you have super high plasma potassium levels because you ate 12 bananas in a row, right? Your body is going to go, whoa, potassium can cause arrhythmia. Not really, it's not going to say that. But it's going to sense the high potassium and it's going to release aldosterone, right? So that you can enhance potassium secretion and excretion to get rid of that excess potassium. Pretty good? So even though this looks complicated, it is a little complicated. I think that you have everything, all the tools to really understand it. So let me put it all together for you. With aldosterone, what are you going to get? Enhanced sodium and chloride reabsorption and enhanced potassium secretion. Okay, that's what aldosterone does. Chloride is actually going to follow the sodium movement because there's more sodium going in that direction. All right. I'm just going to be kind, and I'm going to say, do not memorize any of these molecules. I'm skipping right over it. The only thing that I really want to stress is the difference between creatine and creatinine. Okay? Creatinine is a marker used when you plug it into the clearance equation. You're basically calculating GFR. Okay, excretion, just really briefly. Excretion is basically a coordinated effort between the parasympathetic, the sympathetic, and the somatic nervous system, the motor neurons and skeletal muscles. So just to save time here, the smooth muscle that is surrounding your bladder is called the detrusor muscles, and they're under parasympathetic control. So usually during filling, they're inhibited, but when you pee, when you make the official term is micturition, when you urinate, you stimulate those detrusor muscles to contract. You don't. parasympathetic. I mean, this is involuntary. Also, the sympathetic is controlling the internal urethral sphincter. So it's usually stimulated to contract, right, during filling, but then inhibited or relaxed during urination. But nothing should happen until you voluntarily relax those skeletal muscles. You control urination most of the time. Unless there's something really wrong. Pretty good? All right. So I'm just going to fly through this because I want to save some time for respiratory. Okay. So I think this is now pretty self-explanatory. I don't think I have to spend too much time because we've already kind of, with this is how the loop of Henle basically establishes that osmotic gradient. It's coming up here slowly. Okay. Oh, here's that figure, right? The vasa recta. We talked about this before. The vasa recta isn't organized quite like that U shape. It does surround the that loop of Henle of the juxtamedullary nephron. All right, so this should make sense now. You can actually see how salts are reabsorbed in the thick ascending limb, but water's not allowed to follow. And it's actually reabsorbed more in the beginning of the thick ascending limb than at the top. Okay. We talked about this. I think the main thing here is to realize that the loop of Henle is a countercurrent multiplier because of these active, that's what these red ones are here, active transport properties. But the vasa recta is all about just passive transport, passive diffusion, passive osmosis. No energy is required with the vasa recta, right? Okay. Question?
So for the vasa recta, what's the point of it being like a counter
Current.
changer?
Yeah.
If it's just like going back in.
Yeah. So that is the question, really. That's the question. The question is, why do you even have a vasa recta? Why is it organized that way? The vasa recta is actually important in delivering oxygen to the epithelial cells of the nephron. So you have to bring oxygen and nutrients to make sure these epithelial cells stay alive. Okay, so that's the major thing. But you really don't want to disrupt your osmotic gradient. So if the vasa recta was organized this way, where it was linear and you were delivering oxygen and nutrients to the epithelial cells, but it basically just went into the intermedullary region and then out of the kidney, what you would do is you would be constantly taking up salts and then washing them away, right? So instead, it's organized this way, so it maintains the osmotic gradient. You take up salt, but then you get rid of salt on the way up. Okay, so it's kind of nice, called a countercurrent exchanger to help maintain that osmotic gradient that's established by the loop of Henle. Good question, because that's the heart of this entire concept. Okay? Yeah.
And why do you need to take up salt at all?
Yeah, because these are just passive transport mechanisms. And because it is going into a more hyperosmotic environment, it's just going to naturally take up salt. Can't help it. Yeah. And then as it kind of rounds the bend, it's going to get rid of that salt. Okay? So one thing that I want you to just think about, because I'm always impressed by this, imagine a red blood cell. A red blood cell. It has to go from 310 milliosmoles to 1200 milliosmoles. Think about that red blood cell and its cell volume regulation. It is going to shrink like hell. And then it's going to actually go back up to its original position. But that's incredible. Think about what a red blood cell has to go through. Okay, so I think that's cool. Remember, this is the countercurrent exchanger, and the take-home message here, no energy is required. All right, urea is recycled, and remember, urea reabsorption is controlled by vasopressin. We've talked about that. And these terms, what is the difference between a diuretic and antidiuretic? Steroid and peptide hormone. Pretty good? Diuretic is anything that stimulates excretion of water, usually producing a large volume dilute urine. Antidiuretic, like vasopressin, renin, and aldosterone, all of those are antidiuretics. It helps to promote water reabsorption so that you produce a very small amount of urine that's highly concentrated. Steroid hormones are slow because they usually promote protein synthesis, like aldosterone. Peptide hormones are quick, like vasopressin. Think about this, okay? I'm going to stress this as well. Vasopressin binds to its receptor on the basolateral membrane, and it's G protein coupled. It's GS coupled. So immediately, it's a quick response. Aquaporin channels are just sitting right underneath the plasma membrane. They're already preformed. They're just waiting to be inserted. So you activate that GS pathway, and it inserts those aquaporin channels immediately, and you can reabsorb water. Pretty good? Preformed aquaporin channels are just sitting underneath the apical membrane, waiting to be inserted. Sweet. Okay, so let's go through the 3 intrinsic mechanisms and 4 extrinsic mechanisms. We can do this. I bet I can do it in 10, 15 minutes. But it should be familiar. You guys should see it and go, yeah, get it now, okay? So we're going to go through it, but hopefully this, again, is review. So let's go through those intrinsic mechanisms. Make sure you ask questions as we go along here. Yeah, here. It's up.
Oh.
Yeah, it's in 2 different locations. If you click on the assignment itself, it has the answer key. Or if you actually go to the renal section, you'll see the answer key as well.
Okay.
Yep. I had a student that was like, where are all the answer keys? So I spent some time this afternoon just making sure. I also opened Top Hat, so you can review the Top Hat questions. Yes. Okay. So, oops. I'll give you a short synopsis of each of the intrinsic mechanisms, and then we'll go through the flow chart. Okay. I did have it right. Myogenic control, tubuloglomerular feedback, and then mesangial cells and its control. Myogenic regulation, tubuloglomerular feedback, and mesangial control. Pretty good? Okay, so let's start with myogenic regulation first. Before I go through this, I want to just make it simple. All this is about is if you get a surge of high blood pressure and an increase in flow into the afferent arterioles, that stretch is actually going to trigger some of those receptors, stretch receptors, and there's going to be a compensatory constriction to try to keep glomerular filtration rate fairly constant. That's all it is. high blood pressure, a surge of high blood pressure, stretch of the afferent arterioles, and a compensatory constriction to make sure glomerular filtration rate stays the same. Myo means muscle. It's referring to the smooth muscle around the afferent arteriole. Genic means origin. So the origin of this intrinsic mechanism is in the smooth muscle cell itself. That's why it's an intrinsic mechanism. So now it's a weird flow chart. I agree. But now it makes a little bit more sense. So if you have a surge of high blood pressure, mean arterial pressure, it increases the afferent arteriolar pressure, increases glomerular capillary hydrostatic pressure, increases that net filtration rate pressure, and ultimately increases glomerular filtration rate. So how do you control that? You get the stretch of the arteriolar smooth muscle, constriction of the afferent arteriole, increases the resistance, decreases glomerular capillary pressure, net filtration pressure, and decreases GFR. So this isn't as complicated as it first seemed, right? All it's saying is if you get the surge of high blood pressure and you stretch those smooth muscle cells surrounding the afferent arterioles, they're going to constrict. And it's just to keep GFR relatively constant. Pretty good? Okay. Tubuloglomerular feedback is a little bit more complicated, but we're going back to this diagram. All right? All this is saying is we're going to start right here with the macula densa cells, right? It's detecting the flow through the distal tubule. And it does that. It's actually those sodium potassium 2-chloride cotransporters that are detecting the flow. So, two things. If the flow is too fast or way too slow, there's not going to be many salts that are going to be able to bind to the sodium potassium 2-chloride cotransporter. Again, think about that. If it's rushing by too fast, you're not going to get the binding of those salts to the sodium potassium 2-chloride cotransporter. Or if it's way too slow, you're not going to get very much salt that's binding to that sodium potassium 2-chloride cotransporter. Either way, it's going to send a signal to those afferent arterioles to slow down, right? Let's just say it makes sense with this going too fast. You want to constrict the afferent arterioles to decrease glomerular filtration rate. That's all tubular glomerular feedback is, right? Now, it gets a little wonky, confusing when you think about why would it do that if this was too slow, okay? So imagine you have such low blood pressure. It's so low that the flow through the distal tubule is pretty much plummeted. In that case, you don't want to lose any more fluid to your environment. So you might as well just shut off the system. You're basically not going to filter anything anymore because your blood pressure is so low. Does that make sense? Just shut the whole system down. Okay, so here's tubuloglomerular feedback. Again, these flow charts are a little wonky, but it helps. All right, so what's happening here is you're getting a surge. In this case, it's a surge of mean arterial pressure, increase in afferent arteriolar pressure, increase in glomerular capillary pressure, increase in that net filtration pressure, and an increase in GFR. How are we going to shut that down? It's now being detected by the macula densa cells. Big black box, you know a little bit more about this than the big black box. It's going to detect the flow rate, send a signal to the smooth muscle cells surrounding the afferent arterioles. They're going to constriction of the afferent arterioles, increase in resistance, decrease in the glomerular capillary pressure, which is going to feed back and decrease glomerular filtration rate. Pretty good? Okay, we're just flying through this now. Mesangial cells. Mesangial control. Again, same idea. They're going to get signals from the macula densa cells. The mesangial cells are outside the glomerulus and inside the glomerulus. The whole idea here is you want to decrease glomerular filtration rate. You can do that by closing up those filtration slits or causing what's known as mesangial loops. So you cut off circulation to certain parts of the glomerular capillaries to decrease the surface area and decrease glomerular filtration rate. Does that make sense? Now it seems a little bit easier, doesn't it? Okay. Now, let's see here. Take-home message here. I think this sums it all up. You can have varying blood pressure. I love this graph because this really says a lot. 80 millimeters, I mean, think about that. Usually we have a blood pressure of about 120 to 80 millimeters of mercury. You can go from 80 millimeters of mercury to 180 millimeters of mercury. And because of these intrinsic mechanisms, GFR and renal blood flow, renal plasma flow is just a percentage of the renal blood flow, stays constant in the face of like seriously varying mean arterial pressure. He's a baby. He's a baby. I know, he's so cute. All right, any questions about that? He's trying to learn. He loves physiology. So this is the last of it, and then we're going to go on to respiratory. This is the last of renal, and then we're going on to all right. Okay, what are the vasopressin, aldosterone, renin, angiotensin, aldosterone, and then atrial natriuretic peptide? The Sesame Street rhyme, right? One of these things are not like the other. One of these things are not like the other. Atrial natriuretic peptide is the only diuretic. Vasopressin, aldosterone, and the renin-angiotensin system, they're all antidiuretics. In fact, vasopressin, AVP, is known as antidiuretic hormone. Okay? The reason why atrial natriuretic peptide is a diuretic and the only diuretic is it inhibits vasopressin, renin, and aldosterone. It inhibits all of them. That's why it's a diuretic. Okay, so I wanted to be careful as I go through this. I'm going to come back to this slide. So let's start with vasopressin. How is it that vasopressin is stimulated to release? How does vasopressin get into our system? Okay, it's actually in the hypothalamus. You have osmoreceptors that are detecting the high osmolarity in your blood. Not high volume, right? High osmolarity. It's going to send a signal. The details aren't important, okay? Knowing that this is in the hypothalamus is important, but you don't have to memorize organum faculosum of the lamina terminalis, subcortical organ. Don't memorize that. Know that there's an osmoreceptor in the hypothalamus, and it is going to promote a profound drinking behavior. This is one of the strongest behaviors, physiologically driven behavior on the planet. Drive a very strong drinking behavior, and you're going to get secretion of vasopressin from the posterior pituitary. Okay? All right. Vasopressin enters, now remember, vasopressin is a peptide hormone known as AVP. Arginine is a peptide, vasopressin, right? AVP. Or ADH, anti-diuretic hormone. Vasopressin enters into your system, travels to the distal tubule and collecting duct, right? You have vasopressin receptors on the basolateral membrane. Remember, the blood supply is over here. Binds to its receptor, GS coupled, right? It triggers adenylyl cyclase to increase its activity. ATP is converted to cyclic AMP, increases pKa, and basically what happened is you have these preformed aquaporin channels that are just sitting right underneath the apical membrane. And as soon as this is triggered and GS pathway is activated, these aquaporin channels are immediately inserted into the apical membrane. You already have aquaporin 3 and 4, they're always in the basolateral membrane. But this is the inducible part of this mechanism. Now you have a complete pathway for the reabsorption of water. Okay? Now that completes the pathway for the reabsorption of water. All right, so let me go back to this one. I mentioned it in class, but I want to make sure that you all know what's happening. This is more of a pathophysiological state. I mentioned diabetes insipidus. This is not diabetes mellitus. This is all due to basically vasopressin. Nephrogenic is referring to the nephron. So if you have mutations of the arginine vasopressin receptor, let me just go back to this. If you have a mutation of this receptor and it's not working properly, you have no capability of concentrating urine. You just are always peeing all the time. And basically, you're always thirsty, you're always dehydrated. Okay, so that's a problem. That is called, let me go back here, nephrogenic diabetes insipidus. Why is it called diabetes? Does it have anything to do with insulin? No. It's because you're peeing all the time that all of your solutes, you have a high osmolarity in your blood, right? So even glucose is actually elevated, but that's because you've lost a lot of fluid, okay? So it's called diabetes insipidus.
How do they try to treat that or anything?
Yeah, that's a good question. With a mutation like that, it's difficult. That's a good question. Yeah. Is anybody doing that for their project? It's a good one. I'll look into it and maybe I can give you a better answer. Yeah. All right, so let's think about this from the flip side. Neurogenic diabetes insipidus. Neurogenic. This actually means that there's something that's awry in your brain, right? Neurogenic. Neurogenic diabetes insipidus is a condition where you can't release vasopressin. It can be because you have a head injury. A lot of times head injuries will knock out the capability of you to release vasopressin, and that leads to the same condition, diabetes insipidus. Okay? That's called neurogenic diabetes or central diabetes insipidus. Pretty good? Okay. Does anybody have any questions about vasopressin? These are just more or less review slides, but it really does help you think about what happens in the presence of vasopressin. That's what's happening with blue. And the absence of vasopressin, which is what is happening here in red. So, when vasopressin is circulating through your body, you insert those aquaporin channels and you are able to pull water out. Now, remember, as you move down the tube, you're encountering more and more hyperosmotic environment, which allows you to keep pulling water out, right? So you can actually form a urine, a concentrated urine, as concentrated as the osmotic gradient itself, 1,200 milliosmoles. Okay? But if vasopressin is not present, there's no aquaporin channels, all of that water stays in the nephron and is eliminated. Yes?
How does the urea part of that, like,
Yeah.
how does that tie into this?
So that's a really good question. Imagine if you're pulling all of this water out, you have the potential of disrupting your osmotic gradient in that area. So good idea, pull urea out at the same time so you can maintain that osmotic gradient. Great question. Okay, that's why urea is an important osmolyte in the kidney, so that you don't get disruption of that osmotic gradient in that general region. All right, urea. When vasopressin is on board, urea is reabsorbed. When vasopressin is absent, you get urea excretion. Okay, aldosterone. Let's keep going. I'm a little over here. I thought we would go faster. This tells the whole story. I've actually already talked about aldosterone. Aldosterone is a steroid hormone that increases these ENaC channels. The more sodium that you bring in, it drives the pump. You increase the activity of the pump. You actually bring in more potassium, which is secreted. So let me just sum it up for you. Make it easy. Aldosterone enhances sodium and chloride reabsorption and enhances potassium secretion. Now, one more thing I'm going to layer on. If vasopressin is present, then water is going to follow the solute movement. So you're going to get water reabsorption as well. So aldosterone is actually responsible for sodium and chloride reabsorption, potassium secretion, and water reabsorption if vasopressin is present. So, anybody do the review questions yet? Okay, if you have a tumor of your adrenal gland and you're pouring out aldosterone, right? This happens. You're going to reabsorb so much salt and so much water. This is called Conn's disease. It causes high blood pressure, like really high blood pressure. So you can see again how your kidney and your heart are really coupled together. Pretty good? Okay, aldosterone. Renin-angiotensin. Where is that? Okay. Take-home message here, again, just like with our severe dehydration, in the face of low blood pressure, renin is going to be released. It does this by a couple of different mechanisms. It's actually secreted in response to those baroreceptors in the juxtaglomerular cells, those greenish cells that are surrounding the afferent arterioles in the kidney. They are actually baroreceptors, so when they detect low blood pressure, they secrete renin. Also, sympathetic neurons in the cardiovascular control center trigger renin secretion in response to low blood pressure. And the macula densa cells respond to decreases in flow by releasing renin. Lots of different ways. Again, what you're doing is you're detecting low blood pressure and you're secreting renin. Okay, so here's the cascade of events. Renin is an enzyme that converts angiotensinogen to angiotensin 1. Angiotensinogen is coming from the liver. Again, renin converts angiotensinogen to angiotensin 1. Angiotensin 1 is not the active form. In the endothelium, in your blood vessels, you have another enzyme called ACE. ACE is angiotensin converting enzyme. Okay? And it converts angiotensin 1 to angiotensin 2. This is the active form. That's why ACE inhibitors are so effective in combating high blood pressure. Angiotensin 2 does two things. It causes vasoconstriction to increase total peripheral resistance that ultimately increases mean arterial pressure, getting your blood pressure back up to normal. That vasoconstriction is important in this process. It also stimulates the adrenal gland right here to release aldosterone. And as we already know, aldosterone promotes salt reabsorption and water reabsorption to bring mean arterial pressure back up. Pretty good? Both of these mechanisms. So ACE inhibitors do two things. They actually help with vasoconstriction, decreasing total peripheral resistance, and it inhibits aldosterone so you don't get as much salt reabsorption. All right, and then, as I said before, atrial natriuretic peptide just inhibits all three of them. That's why it's a diuretic. End of story. Those are the four different mechanisms that I want you to know. You can already kind of see that the heart and the kidneys are tied together because the kidneys are really important in water regulation, ion and water regulation, and it's really having an effect on mean arterial pressure. Okay, pretty good. So we got 40 minutes, plenty of time to go through respiratory. I think my throat is actually getting better as I go along here. It's weird. Okay. Any questions? Pretty good? All right, we are just moving along. All right, so I'm just going to go through some of the basics. I think that we have covered this systematically. I actually added something to your oxygen transport. I wanted to make sure that everybody realized, and I said this in class today, that when you look at partial pressure of oxygen in the blood, partial pressure of oxygen, that's a measure of soluble oxygen that's dissolved in the blood, right? Oxygen that's attached to hemoglobin is not contributing to partial pressure of oxygen in the blood. Now, one thing, I just wanted to give you some statistics on this. When you're thinking about the total oxygen carrying capacity, you have to think about both the oxygen that's being transported attached to hemoglobin and the soluble oxygen within the blood. 98% of oxygen that's transported in the circulatory system is transported by hemoglobin. Only 2% is being transported as soluble oxygen. That's why when we were talking about carbon monoxide today, that's why it's such a big deal. If half of your oxygen binding sites are now blocked, that's basically like taking half of your hemoglobin molecules out of your body. It's like anemia, right? So you really compromise oxygen transport and you reduce that total oxygen carrying capacity. That's why we did that exercise today. So I just wanted to make sure that's clear. All right, so when we were going through this, we actually divided this into the upper respiratory tract and the lower respiratory tract. The main thing here is just knowing that air enters into your body through your nostrils and your mouth. You have various sinus cavities, sinuses. You have a hard and soft palate, right? Pharynx is the back of your throat. Epiglottis is here. It's an omega-shaped structure that when you swallow bends down to close off the trachea so you don't get food in your windpipe, right? That's always not fun. I didn't mention this in class, but I'll mention it here. My anti-smoking campaign. My paramedic friend that I always talk about in class, he told me if they come upon a patient out in the field and they have to tube that patient, usually what they're looking for is these pearly white vocal cords. And that's their landmark. They take the tube, they look right for those pearly white vocal cords, and then they can easily tube you if they need to. If you're a smoker, it's gray and dingy, it's very hard to spot. So it's very hard for them to actually find those vocal cords and then basically tube you. My vocal cords are going to be destroyed after this week. All right, so this omega-shaped, the epiglottis and the glottis are here. The epiglottis is the structure that bends down. The glottis is basically this entire area here, and then the vocal cords are within the larynx. And then the trachea is the first tube of the respiratory system. So this is the slide that I told you, please memorize. It's a very easy slide, but I can tell you, I'm amazed at how many people get the conducting zone and the respiratory zone mixed up. Okay? So, trachea splits into two bronchi, and then it continues to branch into bronchioles. Terminal bronchioles are the end of the conducting zone. The conducting zone is all about bulk airflow. That's the conducting zone. Bulk airflow. There is no gas exchange in the conducting zone. This is anatomical dead space. When I say dead space, you should automatically think no gas exchange. The only way, if I'm exercising, that I can change the anatomical dead space is if I put a snorkel in my mouth and keep running, right? Ridiculous. So, just remember, anatomical dead space doesn't change with exercise. The respiratory zone is where all the gas exchange, 99% of the gas exchange is occurring. That includes the respiratory bronchioles, alveolar ducts, and alveolar sac. You have 8 million alveolar sacs within each lung. Okay, so lower respiratory tract. I also mentioned that the right lung has 3 lobes in humans. Left lung only has 2. It's interesting that the heart sits right here. So when you blow up the lungs actually just go right around the heart. It's actually pretty cool. All right, alveolar sacs. Alveoli are the site of gas exchange. It's not quite the grape cluster of grape model. These are really nice scanning electron micrographs that actually show you what the alveolar sacs look like. Okay, so alveolar sacs. Remember, we are negative pressure breathers. So when we take in air, fresh air and stale air actually mix, right? So I talked about bird lungs. Bird lungs have those posterior and anterior air sacs. Takes 2 breaths, takes in air to the posterior air sacs, and then delivers it when they exhale to the lungs. Second breath, air goes to the posterior air sac, anterior air sac, and then exhale out the trachea. The take-home message here is what I really want you to know. Only fresh air enters into the bird lungs. Stale air and fresh air never mix, which is really efficient. Crocodiles also have this, which I think is interesting, because crocodiles are more similar to birds now than they are alligators. Yeah?
How does stale air mix in the human lung?
In the human lung, you can kind of see here, once you exhale, you still have the residual volume. You still have, and extra reserve volume. You still have air left in your lungs. So when you bring in more air, it just mixes with that stale air left over from the previous breath. Yep. So that's why, and this is significant. Remember the partial pressure of oxygen in the air is about 160? In the alveoli, it's only 105. And that's because of that dilution effect. And then in the arteries, it's about 100. Okay? All right. There was something else. Oh, let's remember it's the alveolar type 2 cells that are secreting surfactant. So when you stretch the lung, if you take in a deep breath, you actually stretch those alveolar type 2 cells, secrete surfactant. It helps keep your airways open. And as we learned today, decreases the alveolar dead space. All right. So surfactant is really important. It helps to reduce surface tension by disrupting those cohesive forces right at the air-liquid interface, right at that meniscus. And it increases lung compliance, or the stretchability of the lung. It's easier to open up and keep open. And remember, surfactant it's not secreted until late gestation. That's why premature babies have such lung issues if they're born before 24 weeks. Okay, so this is where I introduced this compliance, just so I do this so you don't forget. Compliance is the relationship between a change in volume over the change in pressure. If it takes an enormous amount of a pressure gradient to just make small changes in volume, that means you have to work harder to open it up, and this is what's called low compliance. If it just takes a little bit of a pressure gradient to make huge changes in volume, that's high compliance. Okay, so surfactant increases compliance. It increases the stretchability of the lung. Now, going back to the lung itself, you all know about transpulmonary pressure now. I just don't want to forget this. If this is the normal curve, this is cystic fibrosis. Cystic fibrosis causes low compliance in the lung. Blue is emphysema. Emphysema causes high compliance in the lung. You would think that was a good thing. It is not. Emphysema is a horrible disease. It's not about getting air into your lung with emphysema. It's all about getting air out. So they have to go through extraordinary measures to get that air out, which is a big problem. Okay. Going back to surfactant, it is a mixture of phospholipids and proteins secreted by alveolar type 2 cells, lowers the surface tension specifically in the smaller alveoli, okay? Makes the lungs easier to expand, but it has a greater effect on smaller alveoli. Surface tension is lower in the smaller alveoli, stabilizing the alveoli. Deep breaths help increase the secretion of surfactant. And again, production in fetal lung occurs in late gestation. So this was the law of Laplace, okay? This is giving you at the top the relationship between aerostatic pressure. Remember, this isn't hydrostatic pressure because this is all about gases. Aerostatic pressure is equal to 2 surface tension, T is surface tension, times the radius of the alveoli. So if there's no surfactant, according to this particular equation, let's just take these 2 examples. Pressure and radius are going to have an inverse relationship. So for the smaller alveoli that have a smaller radius, it's going to have a really high pressure, right? Larger alveoli are going to have a low pressure. The problem is air is always going to flow from high pressure to low pressure, and you're going to get alveolar collapse. So that's a problem. What is that going to do? It's going to increase alveolar dead space, right? Okay. When surfactant is actually secreted, it has a greater effect on the smaller alveoli than the larger. What it tends to do is when you look at the surface tension, now the surface tension in alveolar B is going to be less than the surface tension in alveolar A. And that's going to equalize the pressure to prevent collapse. Pretty good? So it's all kind of, that's why the law of Laplace is important here. But it helps to prevent alveolar collapse. Sweet. And then we had a top hat question. So see, we're actually going through this pretty quickly. All right. 30 mechanics of breathing. I'm just going to hit the highlights. Make sure that you all know. This was a ridiculous animation. It's funny though.
So does that mean like when you're exercising, would you produce more surfactant?
Yes, absolutely. So you're going to be breathing in deeper. You're going to be, and that was part of the homework assignment too. You produce more surfactant. You prevent alveolar collapse. It decreases alveolar dead space, right? And it helps breathing, basically, yeah. All right, so the anatomy. Now you know that there is two membranes that actually encapsulate the lungs. These are the pleural membranes, always on the outer side, just like the pericardium. The outer membrane is called the parietal pleura. The inner one closest to the lungs is called the visceral pleura, just like the pericardium. And inside is fluid. And it helps to cushion the lungs. I use the microscope analogy with fluid in between. If you take two microscope slides and you put a little drop of fluid in between, you can move it up and down like this. But when you try to pull them apart, it's almost impossible. So the parietal pleura is attached to your rib cage. The visceral pleura is attached to the lungs. So when your rib cage opens, pulls the lungs open as well. Does that make sense? But there's two opposing forces. The chest wall tends to bow out. There's an elastic recoil of the chest wall. And the lungs, because they're elastic, they want to collapse. And those two opposing forces creates a subatmospheric pressure in the intrapleural space because they're both trying to pull those microscope slides apart. So usually the fluid in between these two membranes is about -4, right? Okay, so we're negative pressure breathers. I think this is pretty intuitive, but I just want to go through it really quickly. You have respiratory neurons that are innervating these external intercostal muscles. So when they're activated, it triggers these external intercostal muscles to contract and the diaphragm to move down. So what that does is it tends to pull the rib cage open, push the diaphragm down, and it increases the volume in your thoracic cage, ultimately increasing the volume in your lungs. Okay? Boyle's law states that an increase in volume decreases pressure. Right? So you usually decrease the pressure to -1 in the alveoli. Air is always going to flow from high pressure to low pressure. It's going to pull that air in, right? And Ribs move outwards, diaphragm moves down, volume of thoracic increases, air is pulled in. Pretty good? Exhalation or expiration. That's where the nerves relax, they stop their activity. Muscle relaxes, rib and diaphragm return to their original position. Volume of the thoracic cage, the thoracic cavity, decreases. And when volume decreases, pressure increases. The pressure in the alveoli goes to +1, right? Millimeters of mercury. Air is always going to flow from high pressure to low pressure. It's going to actually force air out. In addition, don't forget there's that elastic recoil of the lungs. The lungs itself actually have elastin incorporated in it. So when, just like a balloon, if you blow it up, you're going to have this elastic recoil that's going to help to force the air out. Yeah.
When you like inhale and exhale, does that do anything to the volume of your inhale fluid?
Yes. Okay, so that's a good question. I'm just going to go to this. When you inhale, the intrapleural pressure gets more negative, all the way to -7. And then when you exhale, it goes back to -4. Yeah. So basically, once you pull the rib cage out, you still have the elastic recoil of the lung tube. It actually makes it even more subatmospheric. It's a really good question. Okay. With this slide, all I want you to know is these 3 definitions. Transpulmonary pressure is the difference between the alveolar pressure here in purple and the intrapleural pressure. This pressure right here is going to be 0 minus -4, so it's going to be 4 millimeters of mercury. It is this arrow right here. It is the pressure holding the lungs open because it's opposing the elastic recoil. So the definition, pressure difference holding lungs open opposes inward elastic recoil of the lungs. So the transpulmonary pressure is essentially this arrow right here. The chest wall pressure is actually this arrow. It's opposing the elastic recoil of the chest wall. Remember, naturally, like I said, when during Halloween you take a look at a skeleton, I've always wondered, why does the rib cage look like it's so much larger than a normal person? Again, your rib cage, if you didn't have your lungs attached to your rib cage, it would naturally bow out like that. People that have emphysema have a naturally barrel chest because they've disrupted these pressures, okay? So this chest wall pressure is opposing the elastic recoil of the chest wall. It's technically the intrapleural pressure minus the atmospheric pressure, -4, -0. And formally, it's the pressure difference holding the chest wall in, opposing the outward recoil, elastic recoil of the chest wall. And then finally, the respiratory pressure is the alveolar pressure minus the atmospheric pressure. It's the pressure difference across the entire respiratory system. And just so you know, this is determining airflow. So when the alveolar pressure is -1, then air is going to flow in. If the alveolar pressure is +1, then air is going to flow out. So again, the formal definition is pressure difference across entire respiratory system. Notice it is usually the same as the driving force for airflow. All right, doing pretty well. Do not look at this slide anymore. This won't be on your exam. Boyle's law. Remember, Boyle's law states it has an inverse relationship between volume and pressure. Ohm's law. This will not be given to you on the exam, You should always know Ohm's law. Good walking around knowledge. The driving force is the respiratory system pressure. Resistance and flow. So the driving force equals flow times resistance. This is just rearranged a little bit, so flow is equal to the driving force over resistance. Airflow is a function of the pressure differences between the alveoli and atmosphere divided by resistance. You will be given Poisson's law, okay? You've seen it before, just about the relationships. The significance here is the same as what we learned with the circulatory system. Small changes in bronchoconstriction and bronchodilation translates to huge changes in resistance because of this R to the fourth value. Small changes in the radius of these tubes, bronchoconstriction and bronchodilation, translates to huge changes in resistance. That's why asthma is such a problem, right? Asthma is all about inflammation of these bronchioles and bronchoconstriction. Has nothing to do with alveoli or ventilation. It's all about bronchoconstriction and bronchodilation and possibly inflammation of these tubes, right? Because that can change the radius as well. Pretty good? Okay, sweet. These are two flow charts that are just saying the same thing. Now you can see this chart a little bit easier because you know what transpulmonary pressure is. We've already talked about this being analogous or representing cystic fibrosis, and this representing the blue representing emphysema. Don't worry about pneumothorax. It's kind of fun for me to talk about that in class. It's really morbid. I get it, but it's actually compelling. It gives you an idea and illustrates these pressures within the intrapleural space. But this won't be on your exam. This will be on your exam. Okay, lung volumes. This is the one that I always lecture from. Please know what the 4 lung volumes are and the 4 lung capacities. Capacities will not be given to you on the exam. So you'll have to know what those are. Tidal volume is just when you breathe in and out normally. Inspiratory reserve volume, so think about this. After a normal inhalation, it's everything that you can breathe into your lungs on top of that. That's the inspiratory reserve volume. So I'll say that again. The inspiratory reserve volume is after a normal inhalation, it's all of the volume of air that you can breathe into your lungs maximally. Okay? Expiratory reserve volume is after a normal exhalation, you're right here. Everything that you can force out of your lungs is the expiratory reserve volume. I said this in class. Luckily for you, you still have a little bit of air in your lungs, otherwise they would collapse. The way you measure this is if I took a vacuum cleaner and I sucked out the rest of the air in my lungs. Like I said in class, that would be excruciating for me and horrifying for you. That would be a way to measure that residual volume. Usually what happens is you estimate that using a calculation, height, weight, age, things like that. Okay? So those are the 4 lung volumes. The capacities are this. Okay, here's the definition of a capacity. I better start with that. The definition of a capacity is the sum of at least 2 volumes. Okay? So the first capacity is inspiratory capacity. It's the tidal volume plus the inspiratory reserve volume. That's the first one. The functional residual capacity is the expiratory reserve volume and the residual volume added together. So it's the air left in your lungs after a normal exhalation. Does that make sense? Vital capacity is everything that you can measure with a spirometer. A spirometer is an apparatus that allows you to measure the lung volumes. Residual volume is the only volume you cannot measure with a spirometer. Okay? So the vital capacity is going to be the inspiratory reserve volume, the tidal volume, plus the expiratory reserve volume. Again, it's everything you can measure with a spirometer. And then finally, total lung capacity is all of the volumes added together. So you could also say it this way. It's the volume of air in your lungs after a maximal inhalation. Right? Okay. Sweet. Okay, so, man. I think we can do this. We only have 10 more minutes. Okay, so remember we talked about minute ventilation. I think this is intuitive to everybody because it's very analogous to cardiac output. Minute ventilation is basically tidal volume times respiratory rate. And it's a flow, okay? But I had to talk about alveolar ventilation and hypo and hyperventilation, okay? So this kind of summed everything up. All three of these patients or individuals have the exact same minute ventilation. So it really doesn't tell you a whole lot. If you subtract off the anatomic dead space, right? If you subtract that off, you can see that alveolar ventilation is very different in each of these individuals. So, subject A is panting, right? Shallow breaths can increase in frequency, but they're only bringing in enough air to perfuse the conducting zone. Not great, right? So, have you ever noticed like your dog? To thermoregulate, they will pant, right? But they're only bringing in enough air into the conducting zone, and they're just trying to thermoregulate to cool themselves down. Every once in a while, you'll notice they have to stop and take in a deep breath, otherwise they'll pass out. Right? That's what dogs do. So, A is hypoventilation, not hyperventilation. A lot of people think that just because you've increased your respiratory rate, you're hyperventilating. But if you're only bringing in enough air, shallow breaths, to perfuse or to ventilate the conducting zone, then you're not getting air into your alveoli sacs. This is called hypoventilation. Okay? B is normal, and C is deep breaths. You can see that I could pass out just by hyperventilating, because what I'm doing is I'm actually also blowing off a lot of CO2 at the same time too. Okay, so in this case, I'm hyperventilating. I subtract off a little anatomic dead space, and my alveolar ventilation is very high at 5,100. Okay, so this is giving you an idea of the difference between hypo and hyperventilation, and the real difference between minute ventilation and alveolar ventilation. Okay, so this is also an important one. So think about this. On this left-hand side, this is hyperventilation. I always say, hypoventilation. I always tell students the most extreme hypoventilation is holding your breath. You're not ventilating at all. Till you start turning blue, you can imagine oxygen levels are going to plummet. Carbon dioxide levels are going to accumulate in your body. Opposite is true with hyperventilation on the right-hand side. As you hyperventilate, you're going to blow off CO2. And remember, CO2 leads to acid production too. So this is a way to control your blood pH as well. Alveolar ventilation can change your blood pH within minutes. And then you're going to bring in more O2, so O2 levels are going to go up. Pretty good? So that takes us to our last, basically what we talked about on Wednesday and today. So we talked about this today. Everybody feel comfortable about Dalton's law to calculate partial pressure? Okay. Dalton's law, remember to calculate dry gas, you need to subtract off the water vapor pressure from the atmospheric pressure and then multiply it by that percentage of gas in the air. Okay. I'm going to skip to Henry's law first. This was another part of our respiratory calculations. Henry's law states that concentration is equal to the partial pressure of that gas times solubility. So what you have here is over time, there's no oxygen in the liquid. As you add oxygen to this beaker, eventually it's going to diffuse. It's always going to diffuse from the higher partial pressure to the lower partial pressure until essentially the partial pressures are equal, and then there's going to be no net diffusion. So partial pressure is giving you a better idea of driving force. Okay? And then I also talked about the difference between oxygen and carbon dioxide. When somebody has pneumonia and you have water in the alveoli, it's usually oxygen that's compromised, not carbon dioxide. So people's lips start turning blue, their fingers start turning blue when they have pneumonia because oxygen is having a hard time diffusing across that liquid layer in the alveoli. Carbon dioxide levels are normal because carbon dioxide is more soluble than oxygen is. Okay? All right, so Graham's law, I'm just going to give it to you bluntly, right? Succinctly, is a combination of Fick's law, and instead of the delta C, we're going to use Henry's law, and we're going to plug in delta P times solubility. That's all Graham's law is. It's a combination of Henry's law and Fick's law. So with this last slide, just remember partial pressure, you're just inserting delta P and S instead of concentration. And you just need to know the relationships. We're not doing any calculations. Okay? All right, so I think that we covered a lot of what I was going to cover now. I mean, I'll keep going here. We're actually at oxygen transport. And the main idea here is I kind of set it up to make sure that you knew what hemoglobin was. You all have seen hemoglobin in probably biochemistry, microbiology, biology. Everybody should know what hemoglobin is. I'll just go through it again. It's a molecule that is in your red blood cells that carries oxygen from the lungs to the tissue. It has 4 heme groups. Each of those heme groups can bind one oxygen molecule. So essentially, one hemoglobin molecule can carry 4 oxygen. And it exhibits cooperativity, right? That's why you get this signature sigmoidal curve. What that means is once one oxygen, if a hemoglobin molecule has 4 oxygen molecules attached to it, and you remove one, it's easier to remove the other 3. Okay? Same with adding it. If you add one oxygen molecule to the hemoglobin molecule, it's easier to bind the other 3. Okay? All right, so it exhibits cooperativity. Don't worry about HIF. That's not on your exam. This is important. This is what I was talking about today. Partial pressure, basically, hemoglobin, the oxygen that's attached to hemoglobin doesn't factor in to the partial pressure. So this is a really good example. Here, compartment A and compartment B have the same partial pressure of oxygen. What you're looking at is the dissolved oxygen in these compartments. Once you add the hemoglobin molecule and oxygen binds to hemoglobin, notice that the partial pressure drops. Now you actually have a driving force for the movement, the net movement of oxygen to go to the next compartment until the partial pressures actually equilibrate. Yeah.
So for that curve that was important,
Yeah.
like as it goes up, so think of this saturation.
This curve?
Hemoglobin come up,
Yeah.
partial pressure is also.
So the x-axis is partial pressure. And it's giving you information as to what happens when the partial pressure is high in the arteries and low in the veins. This is telling you something about, okay, well, how much oxygen is actually attached to hemoglobin when we vary the partial pressure. So you can count on, if the partial pressure is 100 in the arteries, almost all of the oxygen binding sites on hemoglobin are going to have an oxygen attached. But in the veins, it's about 75%. Okay, so the curve is actually representing how much, if you think about all of the oxygen binding sites on all of the hemoglobin molecules in your body, right? That would be 100%. Then this curve is telling you what percent of those oxygen binding sites actually have oxygen attached. Does that make sense? Okay. So the take-home message here, I know we're over. Just remember these curves. This is called the Bohr effect. Anytime you have a shift to the right, you're actually facilitating the unloading of oxygen. It actually lowers the affinity of oxygen, and it makes it easier for hemoglobin to give up oxygen. So that includes high carbon dioxide levels, high acidity or low pH, high temperature, and high DPG. Okay, pretty good. That's really the take-home message. Today I introduced the root effect. With the root effect with eels, it's kind of weird. What happens when you increase the acidity is now, not only do you get a shift to the right, you're actually lowering the oxygen carrying capacity. Like you're taking half of someone's hemoglobin out of their body. Like you're blocking half of the oxygen binding sites on hemoglobin, like we talked about today with carbon monoxide. So you're actually lowering the total oxygen carrying capacity, called the root effect. All right, so I can't hold you too much longer, you guys. I would say we did go over carbon dioxide transport today. The thing that I really want you to understand is memorize one of two of those diagrams. So memorize both of them. One of those diagrams is going to be on your exam, a labeling exercise. Okay? Also, yes. Yep, I better show that to you. Remember we talked about the hamburger effect today. This one, or this one?
You want us to know the HB part?
Yes, this is hemoglobin. I would just say I actually named this one carboamino hemoglobin. This one right here. Okay. So, oh yeah, your problem sets are up here. Megna actually graded it today if you want to pick it up. The answer keys are online, so you don't have to pick it up. But Thank you for bringing those.
Which
That's okay.
one is the? I wasn't able to find the second one. Like I've noted down who submitted it so I can grade them.
Okay.
later, but I wasn't sure. I found the respiratory calculations, but I wasn't sure which the other one was.
Okay.
Because there's a into thin air still on there.
Okay, I'll take that off.
All right.
Yeah. Okay, one more thing. I do want you to think about carbon dioxide levels. Like, think about when you're looking over and reviewing, where in your body would carbon dioxide levels be the highest? Right? Where would oxygen levels be the highest? Oxygen levels would be the highest basically right after the blood bypasses the lungs. Carbon dioxide would be the highest right before it enters into the lungs, right? So think about that as well. Peripheral chemoreceptors are another one we talked about today. Remember, they're sensing high carbon dioxide levels more than oxygen, right? All right. Okay, good luck, you guys.
I'll see
Good
you.
luck.
Melissa. Take care.
Yes.
Thank you.
Bye. You too. Good luck studying.
So you said it at the beginning of the respiratory intro, and I think I just
Oh,
zoned
that's
out.
okay.
But you said something was blocked to decrease O2 release or use or something like that. I don't know. I started to like listen back to the beginning of the intro to respiratory.
Okay, so
It was something about like O2 use, like something's blocked, and then your ability to like use it is decreased.
okay.
I don't remember what it was though.
So was I talking about asthma?
Talking about what?
Asthma?
Maybe.
Okay.
Well, no, I think it was before asthma.
Yeah. Got to talk about ventilation perfusion matching as well. So let's just
Well, it's
take a
still
look.
recording right now.
Oh. Stop this recording.